hit the home stretch now. We're almost done. Dr. Hinkle's coming up, and I think Dr. Hinkle's coming up to give us some final thoughts. Yeah, there you are. Dr. Hinkle, thank you so much for your part in making this possible.
Thank you. All right, I'm gonna shorten this down. It's been a long morning. I do want to just make a comment that when you do talk with folks about your experience here, please give a thumbs down to middle-aged men dancing on stage. All right? All right. So I want to just, and I am going to shorten this down a little bit. The main point of this, what are kind of the key mechanisms that scientists are working on, and just to get a broad sense that people are working pretty hard in terms of the clinical trials, things that are happening now and kind of a little bit in the future. So you've seen this before. There are 400 or 500 clinical trials going on at the very least. The upper half, disease-modifying therapies to try to slow down progression of Parkinson's disease at the level of the pathophysiology, and then the bottom part, even more clinical trials in different stages looking at symptomatic therapies to try to improve symptom control. The way the scientific process works is there are basic science studies of molecules and animal models, the dish, et cetera, that come up with ideas that might work. And then they are used in people. Phase 1 clinical trials are very small in size, just testing a compound. Is it tolerated? Are blood levels up, et cetera? And once it passes that, it'll go to a phase 2 level, kind of a medium-sized trial, more people involved, still looking at side effects, blood levels, what have you, but also looking for a clinical effect. And then phase 3 trials are when things are looking pretty good, large clinical trials, lots of people, maybe different doses of the medication, looking for the clinical effect. And when there is a clinical effect, then they'll go to FDA, who will look at it, and that's when approval will be given to a medication to try for symptom control or hopefully soon disease modification. But that's the process. And all the very blurry words up here are compounds that are being used. Why don't we have a cure yet? People have been working on this for decades. What the heck is going on? Well, a couple of levels of difficulty that cause that to happen. The dopamine cells that we've been talking about for years with people in the audience are concentrated in this area deep in the midbrain, and this is a section of the brain like this. So you're looking kind of head on in the substantia nigra. And as a matter of fact, it's an even smaller piece of the substantia nigra at the base and laterally that has the dopamine cells, and it's about the size of a piece of cooked rice on each side of the brain. It's deep in the brain. We just can't go in and take serial samples of it and get biochemical samples and count cells. We just anatomically, we can't get down in there. Anyone's in the room as a dermatologist, we're a little jealous of you because you can take a piece of this and say, what is that? And study it. It's really hard to do in people. So that's an anatomical and size barrier in terms of trying to understand. Also, we really think that Parkinson's is probably going on for 5, 10, 15, maybe even 20 years before it's clinically diagnosable by the tremor, stiffness, and slowness features. So we really, we can't pre-symptomatically take brain biopsies to see what's happening. Most of our pathological data is based on the far right of the screen, where we look at a brain when a person is finished on using it, maybe 30 or 40 years into the course. And then we slice it up, look at it under the microscope to see what's happening. But that reflects the year 40 of what's been going on, and also the death process. So there's we can't really get a good window into what's happening in terms of kind of at the cellular level. Also, it's really, really complex. There are a variety of mechanisms, some of which are shown here, involving subcellular organelles, so little parts of the cells, oxidative stress mechanisms, inflammation mechanisms, and trash buildup mechanisms that all kind of work together and against one another and augment one another. It's just a very complex process that is still being worked out. What I'm going to do is I'm going to take you through each of these kind of pathological processes, and I have one slide for each that has a little bit of specifics that I think I'm just going to not go through because I think we're getting the point across. So a portion of our cells, little organelles inside called mitochondria. Mitochondria produce energy. We breathe in oxygen. Oxygen is run through a course of enzymes within the mitochondria, and energy is produced that allows us to function. So aspects of Parkinson's disease may be from reduced energy for the cell to function, but actually mitochondria do a lot more. And there are a lot of functions that they have that become disrupted in Parkinson's disease, and actually sometimes not just become less active, but actually are kind of turned upside down and become, frankly, toxic. So mitochondria have been a focus of intense research over decades now, but the techniques are getting better to look at them. If we just look at the kind of the upper part of this wheel here, there are several agents that are now in phase 2 clinical trials that work on the mitochondria directly. And as promised, I'm not going to go through them, but there are some agents that in particular, and some of these agents are actually found at pretty high levels in fresh fruit, vegetables, so that might be in the interim way to get them. Oxidative stress has been a point of interest for decades. Oxygen, we need it, but oxygen can also be changed chemically to become reactive and interact with molecules in our cells, modify them in ways that they either don't work anymore or they become frankly toxic. And so oxidative stress has been a key interest point for a long time. People have been studying antioxidants, by the way, for decades. High-dose vitamin E, high-dose vitamin C, glutathione. It is hard for us to measure outcomes in clinical trials because we can't get deep in the brain to see how many cells are surviving, but things have not really panned out so well so far. But there are still 2 agents now in phase 2 clinical trials for disease modification that are antioxidants. Some of them are listed here. Again, common theme, some of our foods. And I gave a talk down the hall in more detail about this, but Mediterranean diet and a lot of fresh fruits and veggies have a lot of biologically active molecules that Mother Nature has made that I think are good to use in the form of lower processed food. But just to make a point that mechanism is still being actively researched. Inflammation, and particularly neuroinflammation involving neurological structures, involving cell types that support the brain cells, is in recent years in particular become a much higher focus level of research for the damage it does to the brain. Taking aspirin, taking steroids, that's not how we approach this therapy. There have been some epidemiological studies of higher use of ibuprofen, lower risk of Parkinson's. So there are phase 1 and phase 2 active trials going on for kind of next level and perhaps more powerful and more directed anti-inflammatory therapies that might have more relevance to the neuroinflammation component of Parkinson's pathology. I've referred to kind of the trash buildup, the alpha-synuclein and other protein buildup that happens when proteins become modified by these other processes that can kind of choke off the cells, make it tough for them to work. We've talked about alpha-synuclein for years. It becomes modified, misfolded, sticky, aggregated together, goes from cell to cell to kind of build up trash, choke off cell function, and make it hard for cells to survive. There are quite a few studies going on in phase 1 and phase 2, and actually one of the agents there is being put together for a phase 3 trial against alpha-synuclein to try to reduce the buildup. And there are molecules in preclinical phase 1, phase 2, and now phase 3 clinical trials trying to block each level of these mechanisms to not allow misfolding, to not allow stickiness and aggregation, to not allow buildup by actually removing the aggregates. There are some vaccinations being tested, and there are actually infusion therapies of antibodies that glom onto these aggregated alpha-synuclein products to try to remove them from the brain. So that's an active area of interest. We didn't go through this in any of the talks today. This was down the hall. But our brains also have, or cells have, mechanisms that detect misfolded, toxic, non-functional proteins and will actually target them to acid pits or organelles called lysosomes. They're put in these little organelles, and the acid digests the abnormal proteins. And then there are shredders that are called proteasomes that will actually identify and shred up these abnormally folded proteins. So there are molecules being worked on to try to augment these mechanisms to clear the brain cells of the trash that it's trying to build up so that only the normal protein is there. A lot of work on environment and genes, environment several decades, and genes more recently. Environment is becoming more and more recognized as important. A lot of epidemiological data about chronic low-level exposure to herbicides and pesticides. More recent work on air pollution, increasing the risk for Parkinson's, and much more recent work on infections, so viral infections in particular, flu and COVID, increasing the risk for Parkinson's disease. And so probably by inflammatory and some of these other mechanisms that I brought up briefly. So, hopefully governments working on reducing pesticide and herbicide use in the environment, trying to clean up the air, and it's a touchy point these days, but masking and vaccinations can really reduce a person's risk for getting these infectious diseases. But that's an active area of research. Genes, in probably the last decade in particular, that's a list, partial list of the main genes that when mutated will cause Parkinson's disease. These genes are being looked at very carefully for what's the mechanism. If this gene is mutated, what happens? All of these pathological processes listed on the slide are downstream processes from these genetic mutations. It's kind of a way to go through the back door to look at the pathology. So, okay, I can't get to the tissue early on, but this genetic mutation is going to cause Parkinson's, so what does it do? Where do the pathways overlap? Are there common pathways that they all go through? Can we learn about that and then figure out how to fix that part of the process to keep Parkinson's from happening? These three at the top are the most intensely studied at the moment. And as you can see, there are, when looking kind of across the board at genetic mutation, disease-modifying therapies, there are phase 1, phase 2, phase 3 trials going on, and lots of preclinical. So that's a really hot area of research. that I think is going to pay big dividends, because I think that's where we're going to find the biochemical mechanisms and the pathophysiological mechanisms that are most critical. GLP-1, glucagon-like peptide-1 agonists are a very, very hot topic. They're on TV commercials, not for Parkinson's disease, but you've probably heard of Ozempic and Rybelsus. I'm not paid by these companies, so I guess I can say it, but I wouldn't know what semaglutide is without knowing it was also Ozempic. But this is a very, very hot topic in neurodegenerative research in general, Alzheimer's in particular, but Parkinson's not far behind. There are phase 2 and phase 3 clinical trials going on for these agents. The phase 3 trial was just published and unfortunately didn't show significant benefit, but one of the phase 2 trials did show some nice response, and that will be going on to phase 3. So a lot of work there. The reason that there's so much interest, these agents have long been studied for reducing the risk of diabetes mellitus. They increase insulin release to try to reduce blood sugar levels and maintain that type of control or lower the risk for type 2 diabetes, but there's also association of high blood sugar diabetes mellitus with PD risk. Also, these agents kind of make us not as interested in things, so we're not as interested in eating, maybe not as interested in using drugs or alcohol. That helps to reduce weight, which also reduces the diabetes mellitus risk and probably the PD risk. In animal models, some of the mechanisms I've been talking about, alpha-synuclein accumulation, neuroinflammation, neuronal cell death, have also been shown to be reduced by these agents, but a lot of the things that have been used in previous human trials have also been found to have benefit in animal models. So we'll have to see where those go. My recommendation to people, there's a lot on the internet. There are a lot of people telling you should use this, you should use that. Some of that stuff may be right, but we don't know a lot about it yet. I can't vouch for things that haven't really gone through rigorous study. I hate to hold people back and be the guy like, be careful, but we really need to try to understand not only the benefits of these agents, but also the potential downsides, and there are side effects to these agents, and scientists want to deliver to you agents that are actually doing something and are safe. So I do like to have some science behind what people are doing. And as I've said a few times, and as you know, people are working really hard. Please support science with your elected officials. Let them know that it's important. These are the only people that are going to be working to help to slow this down or stop it. And I went earlier. I didn't talk this much. I got my 3 minutes back. So that's it. Thank you for your time. I hope it was helpful to you. I want to thank all the volunteers who I forgot to thank last year, but I meant to. Melissa and her team, Jessica and her team, Steve and your group. Holy cow, what a great job on the AV. The speakers and all of you, and we'll let you go for the rest of your Saturday and just watch the kids. Apparently there are a lot of kids out there today. So keep doing your thing, keep having fun, keep living. There's a lot that you can do to help yourself. There are a lot of people around that can help you, and I have been amazed at the strength of the people in this community. I've seen people really optimizing their lives and doing good things and enjoying through tough circumstances that they didn't ask for. So I'm impressed by you folks, and have a good day.